Good afternoon, everyone. Thank you for joining today's webinar. This is Dr. Simi Sugadan. I'm the founder of Product Dev Edge Private Limited. We are building a platform called Maya. It's an AI platform meant for product development, cosmetic product development. The platform is ideally used by formulators, R&D teams, and we help you to accelerate your product launches faster using our resources on the platform. We will be conducting a business workshop close to our product launch. I request all of you to kindly follow our LinkedIn page so that you will know the updates and get to understand what is Maya is all about. Another set of customers on the platform would be ingredients, manufacturers, distributors, and suppliers. So any ingredient that can be used in cosmetics, these manufacturers and suppliers and distributors can use our marketplace and showcase their ingredients for R&D teams. There is a procedure. We will be sending out a business workshop next, I think in a couple of weeks, where I encourage anybody who's involved in chemical or specialized chemical or natural ingredients manufacturing, distribution, or supplier. I encourage you to be part of that workshop because our marketplace will be available for you all to upload your ingredients and showcase. And there is a limited time where you can do this for freely. So please use that opportunity, and I would request you to kindly follow our LinkedIn page to get these updates. Now, without any further ado, let me come to the webinar of today. We have with us Nirmala Ronnie, a very renowned person in the field of microbial stability. She's got over 22 years of experience in the field, and she has worked more than a decade in Unilever and has worked specifically in microbial stability in cosmetic formulations. So without any further ado, I hand over it to Nirmala. Nirmala, take it away. Thank you.
Okay, thank you, Simi. Okay, so good afternoon, everyone, and welcome to this webinar, where we are going to talk about a very interesting topic, cosmetic preservation. Though I'm not sure what what your profiles are, but I have kept it very basic and at an overview level, where I will talk about cosmetic preservation as applied currently in the industry, and also some of the aspirational things. And why I call it a balancing act is because there are a lot of parameters to be balanced when looking to preserve a cosmetic product. And let us start with what the content will be for this webinar. So I will talk a bit about cosmetic microbiology. What are the different types of microorganisms found in cosmetics? What factors, both product-related as well as environmental-related factors, which can cause the cosmetic to be vulnerable to microbial attack. And I'll also talk about the factors which can impact preservation. Then we will move on to look at, what are the considerations which we must keep in mind while choosing preservative, whether it is a natural preservative or a conventional preservative. I will touch upon natural preservative a bit, but I'll not go into specifics. We will keep it at a broader general level. And finally, we will end up with talking about the preservative efficacy testing, how it is done, why it is done, etc. So let's start. So I often hear this, with all the negative PR going on about preservatives, there is there are these questions raised. Why do we need to preserve cosmetics? Do we really need preservatives in our formulation? Because these are chemicals which are harmful, and I think this whole scenario has emerged from the experience we had with parabens, with triclosans, and the likes of that. So it is not just one aspect that they are harmful to health, but also the other aspect of it where people are talking now about antimicrobial resistance due to the biocides present in cosmetic products. While these arguments may have their own benefits, but what we are today going to talk about is whether adding a preservative weighs in or weighs out, or it offsets, the risk or not. So I would say adding a preservative to a cosmetic product which has water or is likely to ingress water is of utmost importance. Yeah, it needs to be preserved, because if not preserved, it can put consumer safety at risk. And that's the main reason why preservatives are put into cosmetic products. Now, if you look at some of the pictures which I have put on the left-hand side, you can see a mold growing in a cosmetic jar, and then some bacterial colonies on another foundation kind of a cosmetic. So it is not just that it can cause consumer harm, but also aesthetically it is not pleasing, and no one who would see such a product is going to use it, or reconcile to the fact that okay, other batches might be all right. So it leaves a lasting impression on them, and ultimately what all of this does is that it harms the business, the brands, and one is the business continuity is affected, as well as there can be huge losses because of other aspects like recalls, and the negative PR generated because of this. Now, we hear a lot about food recalls, but though cosmetic recalls are not as frequent or as many as food recalls, there are still a substantial number of product recalls which do happen. Though being in India, we may not hear too much about it, but in US and Europe, it is quite common, also because they have a very rigorous alert system in place. So if you look at the chart on the left, which talks about, the cosmetic product recalls which have happened between 2014 and 2019, you will see that around 80% of the product recalls were due to microbiological contamination. So that is how, therefore, this topic is of utmost importance in that sense, I would say. So let us also look at some of the major recalls which have happened in the last couple of years. So there was this Nature Truth LLC's lavender body cream which was recalled, and it was contaminated with very, very high levels of yeast in it. Then we had a hand sanitizer recall. This was during the COVID times because hand sanitizers were much in demand, and this was a non-alcoholic product which had to be recalled because of microbial contamination. Now you can imagine this is an antimicrobial product. It is meant to kill microorganisms, but it was not sufficiently preserved. And we will discuss about why this would have happened, and maybe we'll keep it for discussion at the end as to how a hand sanitizer can get contaminated. And of course, the last one is the famous recall which happened for this very premium product, where it was found to be contaminated with very high levels of microorganisms, despite having multiple preservatives in it. So again, this is quite a mystery as to how such a thing could happen. And we will just move on, and we will come back to these examples at the end of the presentation where we can all discuss instead of just me talking about it. Yeah, so I would say that there is no question as to why preservation is not required. Preservation is required for all cosmetic products which may contain water, or even dry cosmetic products where there is a vulnerability to ingress water or moisture and therefore cause microbial growth. Now, what does preservation do? The primary goal, as I said, is consumer protection, but also to maintain product aesthetics and stability. And when I talk about product stability, what can microorganisms do to our products are kind of listed on the left-hand side. They can do much more, but some of the things that can happen is microbial lead phase separation. So if you have an emulsified lotion, for example, microbial activity can cause both the phases to separate out as water and oil. And there can be odor changes because of all the metabolic activity which goes on in the microorganisms. They can produce volatile compounds which can cause the smell to change, your fragrance to change, and it is perceivable by the consumers. There can be color changes. There can be a shift in pH due to microbial activity. It can modify the viscosity of your the formulation. Of course, cloudiness is something which can be seen if your product is clear. So microbial growth produces turbidity and it can cause cloudiness. And of course, like molds, there can be visible growth, which is not very pleasing to the eyes. So the aim of a preservative is to enhance the shelf life and prevent any kind of biological or physicochemical deterioration because of the microbial activity. Now, I'll just talk a little bit about what are the kind of microorganisms found in cosmetic products. It's just an overview. So there are three main types of organisms found in cosmetics, that is bacteria, yeast, and mold. And within bacteria, there are two types, gram-positive and gram-negative. So these two types are simply divided as gram-positive and gram-negative because of the dye intake. That is, upon staining, they take up a dye, and the ones which take up the dye are called gram-positive, and those who do not retain the dye are called gram-negative. So this is a classification which is used in classical microbiology, and it also, kind of helps to differentiate two very classes of bacteria, which are very distinct in terms of their activity and I would say attack on cosmetic products. Their activity may differ widely. So I'll give you a few examples of these, different classes of organisms which are found in cosmetic products. So they are Pseudomonas aeruginosa, Burkholderia cepacia, Klebsiella pneumoniae, Enterobacter gergoviae. These are simply some examples. There is a whole more list. There can be Serratia, there can be Acinetobacter, many more organisms which are part of this list. So this is not an exhaustive list. I just wanted to give you a hang of a few organisms at least. So in the gram-positive side of it, we mostly found Staphylococcus aureus and Staphylococcus epidermidis as the main culprits. And amongst the yeast, there is Candida. There can be others as well, but this is the main main yeast which is found. And amongst the molds, one example is Aspergillus niger, which is also called as Aspergillus brasiliensis nowadays. So as I said, there are many more organisms which can come from different sources, but these are few very prominent ones, and it's good to keep them in mind. Now, where do these microorganisms come from in the cosmetic products? So when we move the continuum from in the manufacturing, in the supply chain, it starts with sourcing of raw materials. So your raw materials can be contaminated. In cosmetic formulations, raw materials can be of two types. They can be inert materials, that is, it doesn't lend themselves to biological activity, but there can also be organic and readily biodegradable materials, which are the main sources of these contamination, and that is why the formulation becomes more vulnerable. Then, of course, there is water. So it depends on the type of treatment the water has undergone. If not treated well or to the extent which is required for that particular formulation, then water can bring in a lot of contamination with it. The other source of contamination is the manufacturing environment itself, the processing. So the main culprits there are, of course, the manufacturing lines. If there are closed lines, closed systems, which undergo CIP, that is cleaning in place, sorry, then at times, if the manufacturing lines are not designed to hygienic standards, they may have some dead ends, and then make themselves vulnerable by retaining some residual product where bacteria can grow, and these are not accessible to cleaning, manual cleaning. So therefore, these form pockets which can keep releasing microorganisms into the product, which can contaminate it. And the second level of contamination in a manufacturing environment comes from the environment itself. So the aerial contamination I'm talking about. Most high-end manufacturers nowadays have aseptic environments, for at least for the places where the product is exposed, say filling, etc. These are under clean room or HEPA filtration systems, but not all manufacturers can afford that, and the environmental contamination is also one of the main causes. And when I talk about environmental aerial contamination, it is mostly mold spores which enter the product. And the third stage, of course, is the packaging material. Though the packaging material is dry, if it is not sterilized before filling, or if it is not clean enough, then it can also bring in microbial contamination. Especially the liners on the caps, etc., may have spores which grow if there is a hot fill and there is some condensation, then those spores can grow and contaminate the product. And then finally, consumer use. Consumer use is one of the major places where contamination occurs, especially in cosmetic products, because one is the direct use, because these cosmetic products are meant to be put on our body, our faces, our skin, hands, etc. And sometimes the design of the packaging is such that they are jars, and we tend to put our fingers inside, or they have to be rubbed on our lips, or the what you call brushes, etc., which are used with the implements used, have to contact the skin as such. So there is a transfer of microorganisms from our body into the product. The second place from where the microorganisms can get into the product is because some of these products are, due to the very nature of their use, they are stored in our say our bathroom shelves, where they are prone to water ingress or moisture ingress. So say, for example, the shampoo bottles, or there can be a body lotion. So those kind of products are very prone to water ingress from the storage environment. And also, when we talk about, though not directly linked to, bringing in the contamination into the product, there are atmospheric conditions like temperature and humidity during transport and storage, which will also determine whether the microorganism which has already entered the cosmetic product will stay as is, or it will grow, given all the optimum conditions it might come across. Okay, so the first three which we talked about, that is until the product has gone to the consumer hands, the kind of contamination which happens is normally controlled by the good manufacturing practices. And when I talk about good manufacturing practices, they may include things like raw material specifications, where microbiological specifications are defined for each raw material to keep the contaminants at bay. They can also include specifications for cleaning in place, CIPs, the kind of sanitizers, etc., to be used, and what level and concentration to use, at what frequency it needs to be done. Then things like environmental monitoring of the manufacturing area. All of those things come under GMP. And there is a very important component of human interface, where personnel hygiene and training plays an important role. Of course, the packaging material also comes under GMP, where packaging materials are bought in clean. So there are packaging material specifications. The COA defines what kind of packing material should be supplied to the unit, and also how to store, how not to expose the packaging material such that they remain in the sterile condition or the clean condition that they have been received. So all of this comes under GMP. So now let us look at where does preservation play a role. Preservation is mainly supposed to take care of the contamination coming in from the consumer use. Yeah, though it does take care of some of the residual contamination which might be left from the product itself, because we don't expect these cosmetic products to be sterile, and neither is there any kind of regulatory requirement for them to be sterile. They may have very low levels of microbial contamination, which is fine, and therefore preservation takes care of this. But as I said, preservation is mainly for the microorganisms entering through consumer use. Now, let us look at the formulation, the factors which may impact preservation. Now, there are a number of factors in the formulation itself which can impact preservation or microbial vulnerability of the formulation. So it is kind of, it plays both ways. It can enhance your preservation, or if not done properly, it may make your formulation vulnerable to microbial contamination. So some of the things which are very prominent are the water activity, the pH, presence of multifunctional ingredients or preservative boosters, as we may call them, which aid in antimicrobial activity of the preservatives. And there can also be environmental factors like the temperature or humidity, etc., which may also play a role. If you look at the picture on the right, if you are a formulator or you work in the cosmetic space, R&D, you would know that if there is an emulsion, the emulsion can be of varied types, and the prominent ones are water in oil emulsion and oil in water emulsions. So it simply means that when we talk about water in oil, it simply means that oil is the continuous phase and water is present in it in the form of droplets. It is a discontinuous phase, and vice versa in the case of oil in water emulsions. Now, since we know that microorganisms grow in water, the ones which are more vulnerable are the ones where water is a continuous phase. Simply because there is more water available for microorganisms to grow. So this is also a formula. I mean, if we look at the microbial vulnerability, water in oil versus oil in water, the one where water is a continuous phase may be more vulnerable. Now, how all these parameters come together, though I will talk in the next slides about each of them in little more detail. Where these parameters play a role in preservation is that they can form a good hurdle approach to product preservation, because they aid in preservation in some way or the other. So it kind of provides multiple hurdles to the microorganism, and it is difficult for the microbes to then overcome all of these hurdles. It can be the water activity. The water activity can be very low, or the pH can be in the range which is not good for the growth of microbes, and similarly the preservative, etc. So there are multiple things which can be put into a product to achieve this, and it is not just the preservative which has to do the function. Now, if you look at water activity, the term simply means that the water is available for the microbes to grow. It is the free water. I'm not going into the chemical definition of it. I'm talking about it in a very simplistic term, that this water activity, what it means is, in a formulation, whatever water is free water, that is, which is not bound by solutes and is present for the microbes to grow, defines the water activity of this formulation. If you look at the scale on the right hand side, you will see a continuum from 0 to 1, where 0 defines that there is no water, and 1 is if it is only water, that is pure water. And in between all the other readings, you see they mark the points at which certain microorganisms can grow. So you would know that you have seen molds growing in bread or in powders and things like that. So molds are the one which need least water. They can grow even with low moisture conditions. So at a water activity of 0.75, most molds can grow. There are certain other molds like the xerophytic molds which can grow at much lesser than this. That is the minimum threshold needed for most of the molds, which includes also the ones which are found in cosmetic formulations. Now, most of the yeast need little more water than that, and at 0.8 and above, yeast can grow. Amongst the bacteria, the gram-positive bacteria are the ones which can grow in low moisture. Like Staph aureus can grow at 0.86. But all the gram-negative microorganisms normally need more than 0.91 to grow. The most notorious contaminants like Pseudomonas aeruginosa or Burkholderia cepacia need much more water at 0.94 or above, only they can grow. So you see that water activity is one parameter where we can control microbial growth. On the right, you see a small table which talks about the water activity of some of the formulations. So this is very generic. It is not that all body creams will be within this range, but it gives you an idea of what is the water activity of these products, and therefore you can imagine what kind of microorganisms can grow. So if we take an example of a body cream, which is an emulsion, it would have high levels of water and low levels of the oil phase. It is a if it is 0.97 to 0.98, everything can grow in it. Yeah, and now these kind of formulations will need a good strategy for preservation, including choice of the chemical preservative itself. While if you look at a lip balm, which is mostly formulated using butters, etc., or oil or wax, the water activity is so low, 0.360, that it doesn't really need any preservation. Yeah, nothing can grow at that water activity. Similarly, if we look at soaps, I mean, very low water activity again. But since soaps are vulnerable to moisture and moisture ingress during use, etc., it would be wise to put a preservative. So, when we look at preservation, we have to also think about microbiological vulnerability of the product and the situations that it will come across during use to decide whether or not to put a preservative and how much to put. Yeah, then the next one is pH. So, I mean, this is quite common. We all know that very high or very low pH are detrimental to microbial growth, and they can mostly grow in the neutral pH range. So, I would say 5. 5 to 8 is the pH where normally most microorganisms grow. We can stretch it a little bit on both sides, maybe 4.5 to 8.5. That is where most microorganisms of cosmetic importance grow. Mm, But this also presents to us another challenge, because if we look at the skincare products, most of them are at a pH of the skin. So it is 5.5 to 7. They are formulated to be in that pH range. So this kind of presents a challenge in the sense that you cannot formulate products with too acidic or too alkaline ingredients. The pH cannot be very harsh, because then it will affect the skin. So these are the different things which need to be taken into account. Now, pH also plays a very important role in preservation when we are choosing to preserve using organic acids. So if you want to have a natural preservative system and you choose an organic acid to preserve, you must keep in mind that organic acids work best when they are undissociated, and their undissociation constant therefore determines how much of acid will be present in the formulation to be active to preserve the product. So say, for example, if you look at the table, there is benzoic, sorbic, and dihydroacetic acid. And you can see what content, what amount can be found at different pH. So benzoic acid has a pKa of 4.2. That is, at 4.2 pH, 50% of benzoic acid is undissociated. Okay. And if it is pH 3, then 94% of benzoic acid is undissociated. But the challenge is we can't keep a skincare product at pH 3. But as you see in the table, as the pH increases, the amount of benzoic acid in the active form, that is undissociated form, decreases. So this is also one of the things to be kept in mind as to how much is eventually available from the preservative that you have put, given all the formulation conditions to preserve your product. Now, coming on to other ingredients. So there are certain ingredients which are called multifunctional ingredients, because though their primary function could be different, they can be emollients, they can be humectants or viscosity enhancers, they can even be fragrances or chelators or antioxidants, etc. Their secondary function could be preservation. Okay, and they could do it by various different methods. It is not necessary that they are antimicrobial in the sense that a chemical preservative works, but they can either modify the formulation to make it more hostile, or they can aid the preservative to get into the microbe by way of either disrupting its cell membrane or things like that. Now, why are these important? Why are multifunctional ingredients important in a product, especially in formulations where we are trying to formulate, in a natural way? This plays an important role, because as per law, in any country, there are only a certain number of preservatives which are in the positive list. That is, say, for example, here I have depicted the EU cosmetic product regulation. The Annexure 5 gives a list of 174 substances now. This was much less, like 58 or something a decade back, and now it has increased to 174. These substances are the only ones which can be used as cosmetic preservatives in a formulation, and nothing other than this, if you want to name it as a preservative. Now, this also comes with very many caveats, like what are the concentrations at which it can be used, and there are various other restrictions to it, including the labeling requirements and things like that. So it is not easy to kind of preserve just with these preservatives, and especially if you want to go say the organic route or the natural route, and you want a COSMOS certification, then this list is much narrow. Now, in those situations, to enhance preservation, these multifunctional ingredients play a major role. So, say for example, essential oils, though they are fragrances, they have good antimicrobial activity. So they actually work as antimicrobials themselves. But there are others, say for example, glycerols. Glycerols are humectants or emollients, and what they can do is they can bring down the water activity. They can find the water present in the formulation, bring down the water activity, just so they make the formulation more hostile for microbial growth. Similarly, there are other, things like skin conditioners, say caprolates or squalene and things like that, which kind of, while giving a skin benefit, also aid in microbial action. So, for example, glyceryl caprolate, what it does is that it permeates the cell membrane of microbes and makes pores so that the other preservatives can enter. It works especially well with organic acids, so it kind of enhances the entry of organic acids into the microbial cells and its disruption. Now, we come to the main topic of this webinar, which is choosing a preservative. Whether it is a natural preservative or a conventional preservative, there are certain fundamental things which we need to be aware of and which we need to keep in mind for us to get good preservation. Yeah. So the first thing is, when we talk about natural preservation, it all depends what your formulation philosophy is. So if you want to go with the natural preservative, you want natural preservation. Natural preservation doesn't mean that your preservative is of natural origin. That is just one component of it. Your preservative being of natural origin is just one component of it. There are other things. So as I spoke earlier, to achieve natural preservation through manipulation of the formulation through other parameters, such that the product is either hostile to microbes or there is less chance of microbial ingress. That is the main thing which needs to be done. That is, hurdle technology needs to be applied. So when we talk about natural preservation, it is a combination of all of that. Now, coming to the formulation philosophy, most of the preservatives which we find today are synthetic. There are few exceptions, of and that is why I again and again keep talking about organic acids or say alcohols, benzyl alcohol and things like that. They are either they are not natural because it is difficult to produce them at that scale from natural sources. So they are nature derived or they are nature identical. Okay, and it depends on where you want to play. You need to choose from this spectrum, which starts from natural, where ingredients are derived from nature itself. They only undergo some physical modification and there is no chemical processing involved, to the right extreme where they are synthetic, which are actually chemically produced. Yeah. So, the things which I have depicted on the right apply to both, whether if you are taking a natural preservative ingredient, still you need to think about all of these things. So, say for example, you are okay to go, somewhere in the middle. You think about naturally derived substances which are chemically processed. Then you can look at a whole range of ingredients, whole range of preservatives which are available there. But some of them do have issues, and I will talk about them when I move forward. Synthetic ones are basically the ones which are in the preservative positive list, and they also have their own limitations and drawbacks. And let us look at all these parameters, and then you will understand better what I'm talking about. So these are some of the things which we need to keep in mind, starting with solubility. And as we spoke earlier, solubility is important because microbes grow in water. Um, uh, I think I have a slide later on that. And then there is optimum pH. Optimum pH means, a lot of preservative have certain pH range in which they are active. And I gave you an example of benzoic acid. So you know that it has a pH range at which most of its active form is available. We need to consider that. Then the spectrum, and when I talk about spectrum, it is about spectrum of activity. Are they active against all the three types of microorganisms, that is bacteria, fungi, and yeast, or not? And if not, do they need to be supplemented by other preservatives which are active against the other type of organisms? Then, of course, cost and availability. So I'm not saying that cost is a very important factor, but some of the preservatives can be quite expensive. Though I would not say that the risk is more of not having your product preserved. If you don't have your product preserved and it goes through a recall, there can be a brand loss, not just financial, but you may not be able to continue your business at all. But preservatives can be very expensive, especially some of the newer ones which we are seeing. They can be very expensive because these are specialty chemicals, or they are directly taken from nature, etc., and it comes with their own cost. And availability, again, you might like a certain type of preservative, but then it may not be available in your region, or the supply may be so limited. Though it is natural, etc., the supply may not be enough for your long term productions, etc. So you need to consider all of those things when choosing a preservative. And cost will also be determined by the concentration of the preservative which is used in your formulation. Something which is used at a very, very low concentration, but it's is expensive, can still, you can still consider it. Those kind of things. Then the next parameter is the safety profile. This is one of the most significant ones. And, though there are regulations in many geographies, especially the developed geographies like the European Union, US, and certain other countries, there are safety guidelines, and the regulatory status is defined by the safety guidelines. This is one of the most important things which needs to be kept in mind. So these preservatives, because of the very nature that they're antimicrobial, they cause harm to the microbes. So they work by various what you call mechanisms of action. So some may disrupt the cell wall, some may disrupt the cell membrane, some may act on the genetic material, and some may even cause disruption in protein synthesis and other metabolic activities. So though the microbes are at a very low, what you call, level of organization in the living organism scheme, still when they act on these organisms and are harmful to them, these can also harm human beings when used at higher levels. That is one part of it. So they may cause issues with our systems. But also there are certain other things which are associated with them. Some can be sensitive to the skin. They can cause skin sensitization. They can cause allergic reactions. And some may even be prone to other systemic effects like cancer production, reproductive toxicity, etc. So those things need to be taken into account. And one of the issues with the new ingredients is that there is not enough information available to take this call, to do the safety risk assessment for those ingredients. And that can be a big barrier to deciding whether or not to include it in your product formulation. The last one is the regulatory status. Not last, I will talk about compatibility also. So regulatory status is also important in the countries where the formulation is marketed. If there are certain regulations, they need to be adhered by. And mostly these regulations are of types like, one is that the microbiological specifications are defined, or microbial limits are defined for ingredients as well as finished product. The second level is that regulatory compliance on safety limits has to be achieved. And third is the labeling requirements. So you need to look at whether you are comfortable with that kind of labeling or not, whether your formulation can meet the regulatory specifications defined or not, and things like that. And finally, compatibility. And this in itself is a very big subject. When we talk about compatibility, not all preservatives are compatible with different ingredients which go on in the formulation. So you need to really check. It can be as simple as the chlorine coming in from your chlorinated water. Suppose you are using chlorinated water instead of treating your water using a reverse osmosis or a UV kind of treatment, then the disinfectant byproducts may interfere with your preservative system. Similarly, if you are using organic acids, you need to be aware of other components which can cause interference with it. Say, for example, I think amines are interfere with the activity of thiazolinones, CIT, MIT, etc. Similarly, there are various such examples, where formulation ingredients can cause issues and make the preservative not work at its optimum. Okay. So as I spoke about them,
just 1 minute. We have 10 more minutes for the presentation, 12 more minutes actually.
Okay. So what I'll do is, I think I've spoken about these things already. I'll just skip these 1 or 2 slides so that we have some time. I've kind of, spoken about all of this. So as I said before, natural preservation is a combination of not just the preservative, but the hurdle technology applied, the preservative boosters or multifunctional ingredients which are present in the product. Now, when I talk about the balancing act, what is it? What is it actually? So as we saw that there are a number of parameters, number of factors which are in play. And to balance all of this, either by way of packaging so that it is not accessible to ingress, inserting your hands, rather using a pump instead of a jar, or choosing the right preservative with broad efficacy, which will be effective in your formulation, or it could be the hurdle effect designed in the formulation. All of this together is what will achieve optimal preservation, and this can only be done if you do a microbiological risk assessment for your the product formulation, and you build in these things by design. So this approach is called safety by design. Now, finally, it is the preservative efficacy testing. The preservative efficacy testing is also called challenge testing, and this is a very simple procedure. And why it is performed is, despite all of this, I mean, you have chosen a good preservative considering everything that I spoke about, but still some formulations, some preservatives fail. So to make it foolproof, to make sure that it really works in your formulation, and it can differ formulation to formulation, each preservative will behave differently in different kinds of formulation, it is necessary that you do preservative efficacy testing. In certain geographies like EU, etc., it is mandated by law itself, but in other geographies, though it is not mandated, it is best to do a preservative efficacy testing. Now, this is simply a laboratory test where, which is done to evaluate effectiveness of your preservative, especially considering the consumer use scenario, whether through the shelf life of the product, your product will remain preserved or not. So there are various methodologies available, and including the PCPC in US, which is defined by the Products Council, and the USP methodology, and even the ISO methods are available. Here I am just trying to tell you, how this test is done. So different types of microorganisms, either alone, that is singly, or in a mixed inoculum, are inoculated into the product. Okay, the finished formulation. When I talk about the formulation, it is your finished product. It has to be in your finished packaging material, inoculated, and then at certain intervals of time, these products are removed, a sample of the product is removed, and then it is put through a microbiological test where you count the number of surviving microorganisms. So it simply tells you whether your preservative was active or not and how active it was. So when you the normally how you do this test is that at day 0, all these different types of microorganisms are inoculated at a very high level, at a concentration of 10 raised to 5 to 10 raised to 6 CFU, 6 log of microorganisms, and then a 0 day sample is taken to understand at the initial time when you inoculated into the product how much was the microbial concentration, and then it is monitored over a period of time of 28 days, and at different intervals of time, that is 7 days, 14 days, 28 days, depending on the protocol you are using, you draw out sample of the product and check for microbes, and from there you can understand whether the ones which you have inoculated have actually died, or they have stayed as such, or what has happened to them. So that will define whether your preservative was effective or not. And all of these tests also define some acceptance criteria. So the one which I have put in here is actually from ISO method, where a minimally for bacterial contaminants, a 3-log reduction is necessary at 7th day, and there should not be any increase going forward. That is, when you test at 14 and 28 days, it should have remained either at 3-log or less than that. For yeast, 1-log reduction is required, but for mold, the requirement is little less stringent. It talks about that there should not be any increase from the one which you have inoculated, and after 14 days, it should have at least gone down by 1 log reduction. Yeah. So this is all, and I would just want to leave you with this thought. This was this cream was found in one of the archaeological sites, and this cream is believed to be 2,000 years old, but it is still intact, and it was found with finger imprints and all. It had no microbial contamination, and I wonder what preservative they used. So yeah, that's all, and thank you. You can contact me at these coordinates, either connect with me on my LinkedIn, or you can write to me at this email, nirmala.ronnie@codexscholar.com. Yeah, and now I'm open to questions.
Thank you, Ronnie. Thank you, Nirmala, for the wonderful presentation. I would like to pitch in some things that you can understand using our platform. So with Maya, you will be able to see some of the aspects of a preservative, such as, I'm just going to share my screen. I won't take long. Is my screen visible? Yeah. So, for example, Nirmala was talking about sodium benzoate and those kind of chemicals which are used as preservative. So, one of the aspects of compliance or safety aspects that Nirmala was saying is, for example, the multifunctional properties of a preservative, and the safety aspects of the preservative, what kind of acute and chronic reactions that you will encounter, that you can find on Maya. And if there is any regulatory threshold limits, which Nirmala was talking about, you can get those information over here. So this is one part of our platform where you can do these ingredient research or your preservative research and understand the profile of the preservative in a detailed manner. And we follow science-backed result information only on the platform, so you can be assured about the reliability. Now you can, the floor is up for questions. I would also request our the people who have joined here to fill our feedback form. So part of the question is what kind of information you want in terms of your next webinar. So we would like to hear from you what you want us to work on. So please fill the feedback form. Okay, Nirmala, there are a lot of questions on the chat box. I'm going to read out some of them. So Saina is asking, please share options for odorless natural preservative.
Okay, Shaina, what I would suggest is that I don't want to really, patronize any particular ingredient or supplier here. But what you can do is you can write to me one-on-one and we can discuss about this. Yeah, there are a few options available, but not necessarily they will be organic or natural if you would like, but other than benzyl alcohol, I think all these there are organic acids which are kind of they will not cause any kind of odor as such.
Okay, Aishwarya Darve, how do we modify preservatives action against gram-positive and gram-negative bacteria? I think Nirmala has covered with that design model. I think, Nirmala, do you want to add any more?
Yeah, I just want to say that you cannot modify a particular preservative's action, but what you can do is you can supplement it. If it is not active against a particular type, then you can supplement it with another preservative which is active for that particular group of organism.
Okay. Saina again, which phase of formulation we should add potassium sorbate and sodium benzoate? That's one question. And the second is, do we need to heat it to dissolve fully?
Yeah, so see, when I talked about solubility, solubility is also quite a dicey thing. So if you are adding, say, benzoic acid, in its acid form, it is less soluble. But in its salt form, that is sodium benzoate, it is more soluble. So that is one option you can think. And the other question which you raised is about where should we add? And it's a very important question, because what happens is benzoic acid has a tendency to move into the oil phase. Okay, it either goes into the oil phase or it is at the interface. So it is essential, the sequencing or step where you add it is very essential. So you should start it at the start of production, in the water phase, so that it remains in the water phase and not really shifts into the oil phase.
Okay, Gayatri Ganeshan wants to know, microbial surfactant is a multifunctional agent, has secondary function as preservative. How do you see this microbial surfactant in commercial application?
Yeah, so many of these are now being used. And they, as I said, they are one of the preservative boosters, the strategies which is being used. So yeah, they can be used provided they have a good safety profile and their regulatory approval is there. So you can use them.
Okay, Anamika wants to know, if we are going to put more than 2 preservatives in a formulation, how do we understand what dosage are going to be the best for the preservation?
So see, most of the suppliers give you the dosage at which they are active. But also, they are also defined by the regulations. So there are maximum limits defined. Now, it is a double-edged sword. Though there are maximum limits defined, you don't want to use it at the highest level. But if you use it at very low level, there is a chance that it can cause antimicrobial resistance development, and there can be some microorganisms which can become resistant in your manufacturing lines, and then that preservative may not work for your formulation in the longer term. So there has to be a balance between the two approaches. You cannot use very low, you cannot you have to be within the regulatory limit or the safety limit which is defined. So you have to choose as per your challenge testing what works best. If you're using 2 or more, all of them have to be within the safety limit defined for each of them.
Okay, Mayud Ahmed wants to know, can you share the EU document which defines preservatives approved to use?
Sure, I can send you a link.
Aishwarya again, how to modify preservatives action against gram-positive and gram-negative bacteria.
I think we
Yeah,
answered
she
the
asked the
question.
question again. Yeah. So it's been answered, Aishwarya. Do we have any number or specific strains that product should be tested before commercialization?
Sorry, so can you repeat that again?
Do we have any number or specific strains that the product should be tested before commercialization?
Yeah. So these protocols which I mentioned, that is the ISO or the, some people prefer to use pharmacopoeia or the PCPC methods, have defined strains. Now, those are the standard methods and the standard defined strains, but a lot of corporate organizations, which are larger organizations, what they actually do is they develop their in-house methods, wherein they use the strains, the microbial strains, which have either come from product recalls, the contaminated products, or from their manufacturing lines, etc. And they know that their product is vulnerable to those, so they try to include those in the challenge test as well.
Okay, I think that's all with the questions. Nirmala, I have one question.
Yeah.
So when we are selecting a raw material, let's say a preservative from a raw material provider, from a manufacturer or a supplier or a distributor, you said that also brings in contamination, right? So apart from the GMP, when the supplier is bringing the chemical or the natural preservative, is there any reports or certification that we can look where we can understand the whether the product is sterile or microbial stable? If
Yeah.
it's a specialized chemical where there is a mixture, I don't know whether there is a preservative. Oh, yeah, there is, right? With a mixture of ingredients in it to cover a broad spectrum. What all documents, apart from the GMP aspects, should we look at a supplier's portfolio in terms of the ingredients?
So see, normally all the ingredients come with certificate of analysis, and microbiological specifications are part of most of them. And when I say that, I'm not talking about preservatives alone. I'm talking about all the ingredients. Yeah. And that is enough, because most of the suppliers define for the geographies that they market their ingredients in, what is the limits to be met, the microbial limits to be met. So it is different. Mostly, I mean, overall the limit is divided a little bit different for two categories of products. One is the ones which are used in baby care products, or for the products for eye care, or which can come in contact with mucous membranes. Okay, the limit for that is little bit stringent. So for example, in the US, PCPC defines that the microbes should not be more than 500 CFU per gram of the product, and gram negatives, that is the pathogenic microorganisms, should be absent per gram. For all other products, it is little more lenient. The limit, I think, is 1,000 per CFU per gram, and yeah, the limit for gram negatives or for pathogenic organisms remains the same for both categories.
Okay, Aradhana, you can ask your question.
I'm sorry, I'm not able to put my camera on. I hope it's okay.
That's okay. That's okay. Okay, there is one more question. What is the expiry period of preservative? If my that is one question. If other ingredients expired after 3 to 4 years, do preservatives work too long? Interesting.
Okay. Who has asked this question? That's a very interesting question.
Yes.
And
Nizam Khan.
okay. So, see, my last slide should answer that actually. You saw that ancient cream which is 2000 years old. I wonder what preservative was there. So, rather than looking, but okay, what I'm trying to say is that you don't want a product which is 10 years sitting on the shelf. You don't want to use that kind of a product, right? So rather than looking at how long the preservative can last, you need to look at what is the shelf life, intended shelf life of your product, given all the ingredients and your formulation system, how long it can last. So that is why stability testing is done and preservative efficacy is done to check, through that shelf life, whether it is reasonably preserved or not. So if you are saying that can my preservative last for 4 years, it will all depend on how many microbiological insults it has gone through and how much of it got used up. Okay. So yeah, I hope I answered your question. Did I?
Yes, he says
Okay.
yes. Okay. Aradhana again wants to know in Lil Earl cleanser had preservatives, still it was recalled because of microbial contamination. Can you explain this?
Yeah, yeah. So this particular product actually had 4 preservatives in it. It had 2 organic acids, it had phenoxyethanol, and I think it also had some preservative booster. Still, it was contaminated. So during the investigations, they couldn't really pinpoint it to the preservative loss. It was more because that product was diluted while getting filled up. So it was a problem in their line, and some of the products which got filled up were diluted, and therefore the preservative was not effective against that. So that was an issue.
So
But
is it a batch problem or
yeah, it was a batch. It was a batch problem, but that batch size was quite big, so they couldn't really separate out the products, and the entire thing had some 15,000 units had to be recalled.
okay. Okay. Shravani wants to know what kind of effective preservatives are to be used in under eye cream or eye cream formulations, since very low amount should be used.
Yeah, see, I'll have to look into the formulation to really advise you on that. Sorry, I can't just offhand say. But you can look at some of the information which are, approved for under eye care in the regulatory space. In the
Yes.
positive list, you can find those preservatives.
Smita wants to know what is common between preservative in food and cosmetics.
Ah, okay. So see, the major difference between a cosmetic and a food formulation is that a food is completely biodegradable. Everything, all components can be used up by the microbes, while the cosmetic formulation can be a little hostile as well because of some of the inert microorganisms. So the level of preservative which is required in a cosmetic from that sense may be lower. But there is another issue with cosmetics. Most of the food products undergo heat processing, but not all cosmetic products may undergo heat processing, or they may only partially undergo heat processing for one particular phase, or things like that. So that is an issue. So, there is not just one answer to it. The formulation on the whole, the process on the whole has to be assessed to decide on the preservation.
Okay, I think we'll take only one more question because we have overshooted the time already.
Sure.
Okay, there is no more questions. So I think we'll stop here. Is Radhika Solanki here? She was raising the hand during your presentation. If you are still here, you can ask the question, Radhika. I don't know whether she's still in the meeting. Okay, I think she's not here. Yeah, so I think we should end the session. Thank you, Nirmala. It was a very insightful session. I hope the people who attended had gained a lot through this. I request all the participants to please fill the feedback form, help us understand what more you want to know in terms of your R&D requirements. We will try to see whether we can come with those topics for our webinar, and request you to please follow our LinkedIn page so that you can get the updates on time and be part of our network. Thank you.
Thank you.
Thank you, Nirmala. Thank you very much. Wonderful
Thank you.
session.